Welcome to lesson 2 of your course on the inflammatory process. This lesson is going to cover some of the specialized cells that are involved in the inflammatory process. Okay, so we took a look at our blood vessels and looked at the anatomy of them. Now we're going to start looking at our specialized cells. So, the first one we're going to talk about is our natural killer cells. So, the role of natural killer cells is to sense whether another cell comes from the self or not from the self. Every cell has different markers on it that will identify whether it came from the body or not. So, when a natural killer cell encounters another cell and it senses it as not a self cell, it will do a couple of things. One, it releases perforin and granzyme B. These two chemicals cause cellular death, which is also known as apoptosis. What it literally does is it shoots holes within that other cell and causes that cell to die. Another thing that it does is releases interferon. Interferon is a chemical mediator that does a couple of things, including activating macrophages and activating mast cells, in which those two are more specialized cells that will help aid us in the inflammatory process. Another thing that happens with our natural killer cells is they themselves will release TNF alpha, which will also activate macrophages. It's another chemical mediator that is released in this inflammatory process and it activates our macrophages. So, here is our role of our natural killer cells. They sense when a cell belongs to the body or when it does not. That could be in the form of cancer or of a toxin or bacteria, or in the case of a damaged cell, it is knocked off that receptor in order to notify and mark it as a cell of the body itself. And it recognizes it saying it is not a self cell. And so it ends up instigating this inflammatory process. So, taking a look at mast cells. So, when mast cells, another one of our specialized cells, are activated, they go through a process which is called degranulation. And I actually have an awesome video that I'm going to show you right here of what degranulation looks like. So, here's our mast cells. And as they are activated, they start to break down into tiny little granules. And they, in the process, are releasing a bunch of different cytokines or chemical mediators. So some of these chemical mediators that it is releasing are histamine, heparin, TNF alpha, and interleukin 16. So histamines, I'm sure you guys have all heard of histamines, right? And when we are experiencing allergy symptoms or cold symptoms, we like to take what are called antihistamines. Well, what do antihistamines do? They block histamine receptors. What happens when histamine comes in contact with histamine receptors is it actually will take all of the cells within the body and start to spread them out from each other. So it creates these intercellular clefts. These are gaps in between all the cells that are lining the endothelium, which are lining the blood vessels. Okay, so it takes all the cells that are lined up on the blood vessels and it starts to spread them apart from each other and it creates the gaps that are in between it. Well, mast cells also will release heparin at that same time. And what heparin does, it actually blocks us from being able to clot those gaps. So histamine creates these gaps in between, and heparin makes it so that we can still continue to have those as open gaps. So that's going to allow for our fluid to be able to start leaking out through our blood vessel walls into the dermis, and also is going to allow for those white blood cells to be able to exit the blood vessel as well. Now, a couple other things that mast cells also release are TNF alpha. And if we remember from the last slide, our TNF alpha activates macrophages. And then it also releases interleukin 16, which will activate and attract T cells to the area. It's another specialized cell that we have within our inflammatory system. So it's activating and attracting more cells to help join in all of this inflammatory response to try and heal the body. All right, so our macrophages are a specialized cell that really does a brunt of the work when it comes to cleaning up all of these damaged cells and trying to repair things. So macrophages originally started as monocytes, which is a type of a white blood cell when it was within the bloodstream. And what ends up happening is they end up releasing out of the bloodstream through those gaps that we have created, and then they differentiate into what are called these macrophages. It is a phagocytic cell in which the one main role of phagocytic cells are to eat, to consume, right? Interestingly, when we looked at the previous slide and we were looking at mast cells, we used to think that these were phagocytic sites as well. And so they used to think that these cells masticated or chewed up other cells. And that's why they were called mast cells, for masticating cells. However, they turns out that they were wrong. And they actually are not phagocytic sites. They actually just degranulate and release these different chemicals. But as macrophages, they are phagocytic cells. So they do consume and eat up debris within the area. During this process, they also release a variety of different chemical mediators. And the two that we're going to really look at is TNF alpha, which activates macrophages. So interestingly, macrophages will actually activate themselves through the release of TNF alpha. So the more activated macrophages are, the more TNF alpha they will release, which will then in turn reactivate them even more. There are many different interleukin cytokines that we have. Interleukin 1 is also released from macrophages. And what that does is it activates our endothelial cells. And we'll talk about endothelial cells and what their role is. But first, let's talk about a recap of looking at our phagocytes and natural killer cells with this awesome video.
The first defensive cells on the scene are your phagocytes. Their name literally means to eat, and like Pac-Man, they indiscriminately chase down intruders and gobble them up. And they come in a few different varieties. First, you got neutrophils, which are the most abundant type of your white blood cells. They kind of self-destruct after devouring a pathogen, and in fact, you've probably seen piles of their little dead bodies, because that's what pus is made of. But the bigger, tougher phagocytes are the macrophages. They're derived from monocyte white blood cells that have moved out of the bloodstream to occupy tissues, and some are free types that patrol tissues looking for creepers, while others are fixed, attached to fibers in specific organs, devouring anything suspicious that passes by. So when a macrophage, in say the finger I just cut slicing a bagel, sees a new bacterium coming along, it snares it using cytoplasmic extensions, reels it in, completely engulfs it, and essentially digests it and spits the rest out. And unlike neutrophils, it can do this over and over again, like a boss. But not all your defense cells are phagocytic. You've also got cells with what is by far the awesomest name of any cell in the body, the natural killer cells. You can call them NK cells if you want to, but like, why would you do that? Anyway, these tiny assassins patrol your blood and lymph looking for abnormal cells and are unique in that they can kill your own cells if they are infected with viruses or have become cancerous. How can they tell? A normal, healthy human cell contains a special protein on its surface called MHC1, or major histocompatibility complex, but if it's infected, it stops making that protein. And if an NK cell detects a defective cell, it doesn't swallow it whole like a macrophage. It pokes it with an enzyme that triggers apoptosis, or programmed cell death.
So taking a look at our endothelial cells. Now remember, this is the lining of our blood vessels. And just for this diagram, so you understand, we are looking at the inside of a blood vessel in this area, and then this is the dermis. And I just want to point that out because it's flipped upside down compared to what we've been looking at in our other pictures that we've seen in this section so far. So, in a general sense, what's going to end up happening is endothelial cells, when they become activated, they will release some substances. One of those is interferons. Interferons will in turn activate more macrophages. So, we've got macrophages that are activating endothelial cells, and then when the endothelial cells are activated, it will in turn go back and activate more macrophages. It's going to also help to call these different cells, the monocytes and the neutrophils, to the area. So, what ends up happening is through adhesion molecules, including vascular cell adhesion molecules, as well as ICAMs, which is intracellular adhesion molecules, not listed on here, but these will start to recruit circulating monocytes and neutrophils that are located in the bloodstream and pull them towards this area. So it kind of like puts out a little hog call that says, hey, we need more help over here. Please start coming over here. Monocytes and neutrophils will start to come to the area, as well as they release substances to actually walk these cells down the lining of the blood vessel wall until it encounters a break within the endothelial wall. This is called margination. When we have this monocyte or the neutrophil is walking down, slowing down to come in contact, and it binds to this lining of our blood vessel wall, and it kind of looks like a gear as it's just traveling down until it ultimately comes into a cleft within the endothelial lining of our blood vessel. When that happens, our neutrophils and our monocytes can actually diapedese, or it's also called extravasation, in which they come from within the bloodstream into the dermis. At this point, neutrophils will now become phagocytic cells and start consuming all of the inflamed and damaged tissue in the area. And our monocytes will actually differentiate or convert and transmorph into macrophages. And so it's a way for us to call more macrophages to the area, as well as our phagocytic neutrophils. All right, so let's take a look at this just a little bit more in depth and ask ourselves, so how do these white blood cells actually get into the area, get called to the area, and end up getting into the intima where we ultimately need them from the blood vessels themselves? So it goes through this process called margination. So what ends up happening is we then, through endothelial cell activation, will activate selectin and integrin. So you will see selectin that will actually appear here and is activated, right? These little green bubbles that we have right here along the wall of the endothelial lining, right? Along the wall of the blood vessel. And also, there's selectin receptors that are activated on our monocytes and on our neutrophils. In turn, integrin is also activated, in which we have integrin that is located on the monocytes and neutrophils themselves, as well as integrin receptors, or we can call them vascular adhesion molecules, that are here, and the integrin will bind to these. So we call these cells into the area. They come and they start to slow down by acting like Velcro as they start to attach onto, from the selectin attaching onto the selectin receptors, and the integrin attaching onto our adhesion molecules, and it walks like a gear down the wall of the blood vessel, right? And it starts to elongate its way out. It starts to slow it down so that they're not just floating by with the blood going past, and instead they're actually slowing themselves down until they can finally walk down and find a break in the system. When we have this gap occur because of histamine, and we'll also take a look at a few other things that cause this in just a moment, that we can actually have this gap where the monocyte and the neutrophil will diapedese and will release into the tissue itself, into the intima. So we're going to take a look at just kind of putting this all together and starting from the beginning and walking through to where we are right now. And it's also going to introduce us to a few new chemical mediators that also aid in the inflammatory response, including bradykinin and prostaglandins. And you'll see how these also play a role in the inflammatory response.
Inflammation is a critical innate defense in the war between microbial invaders and their hosts. Microorganisms trigger inflammation when they're introduced into the body and begin to grow and produce compounds that damage host cells. Resident immune cells called macrophages may wander into the infected area and engulf the invading organisms. The macrophages digest the material and in this way help clean up the infection. In response to the infection, macrophages release a number of chemicals, including those called cytokines. Cytokines are small proteins that regulate the immune response. Some diffuse into the vasculature and stimulate endothelial cells to express specific receptors called selectins. Selectin binds to carbohydrates on the surface of neutrophils, snagging the cells as they flow by in the bloodstream and slowing them down, such that they roll along the endothelium. Inflammatory signals trigger these rolling neutrophils to express adhesion molecules called integrins on their surface. The integrins lock onto adhesion molecules called ICAM1 and VCAM1 on endothelial cells. The tight adhesion stops the rolling, and the neutrophils begin to stretch out along the endothelial surface. Damaged tissue cells in the area of inflammation release bradykinin, a 9-amino acid polypeptide that helps loosen the tight junctions between endothelial cells. Neutrophils can now initiate extravasation, in which they squeeze through the loosened endothelial wall and into the tissues, where they can help macrophages attack the invading microbes. Bradykinin molecules also bind to other cells of the immune system called mast cells that are in the area, causing them to release histamine. The histamine further loosens the endothelial cell junctions, allowing more fluid and cells to move out of the capillaries. Bradykinin, when it attaches to capillary cells, induces the cells to synthesize prostaglandins. Prostaglandins stimulate nerve endings, causing pain, which draws awareness to the infected area. The 5 cardinal signs of inflammation are redness, warmth, pain, swelling, and altered function at the affected site.
Excellent. Well, this concludes lesson 2 of the inflammatory process. Make sure to join us for the next lesson, in which we will start discussing some of the chemical mediators and go a little bit more in depth on these chemical mediators that can be found throughout this process. Make sure you've completed your assignment down below. You can review any of your questions and make sure to press submit.